couple brief comments. Industry is very different from basic science research as far as what they're open to be sharing with. They do work for companies, with the exception of Eric, of course. They do work with companies with proprietary knowledge and whatnot. So they may not want to answer all of your questions or answer them to the full degree that you might want. That doesn't mean they're avoiding you. They're just trying to stay employed by their employers. So please realize that. I have worked with all of these companies up here. They're exceptional individuals. And they very much care about you and the families, and they really want the best for our field. And Rob and Ann have known each other for a while, and I tell them, no matter what the results of the study, the Ionis study is, or any of these studies, we should see that as a success. The fact that we have industry even interested in our disease. So thank you for that. And regardless of what the outcome of these studies are, we thank you. So I guess we will go in order of what the slides are. So would you like to start?
All right, thank you. Thank you very much. Thank you for taking the time to be here today. My name is Kiyoshi Arita, and I am the project manager for NPC-31 at the Nobelpharma in Japan. And Nobelpharma is relatively small company with Tokyo head office and subsidiaries in US, Germany, and China. We have about 350 employees. Our corporate mission is to contribute to society by providing critical but neglected pharmaceuticals and medical devices. I would like to share recent research development on our project, NPC-31, a potential treatment for prion disease, such as Creutzfeldt disease, CJD. CJD is caused by prion proteins in the brain that undergo a structural transformation from their normal form into abnormal disease-causing form. These abnormal prion accumulate and destroy nervous cells. At Gifu University in Japan, a research team led by Professor Kazuo Kuwata has been working on the potential treatment using a specially designed molecular chaperone called NPC-31. NPC-31 is engineered to attach to normal protein and protect them from converting into the harmful form. NPC-31 specially bind to the hot spot, probably this slide showed. And a hot spot of normal protein, prion protein, PRPC, inhibiting its interaction with abnormal prion protein, PRPSC, and stabilize its structure. This development was made possibly through the use of the advanced image technology, especially NMR, that is a nuclear magnetic resonance spectroscopy, to analyze the prion protein structure at a molecular level. This research team began by designing the NPC-31 compound using computer simulation and combined molecular dynamics and quantum chemistry calculations. This approach helped predict which molecular structure would most effectively bind to the normal prion protein. Over 200 candidate compounds were synthesized in the laboratory and screened for their binding ability. NPC-31 emerged as a most promising compound showing strong binding affinity to key region of the normal prion protein that's critical in preventing its structural conversion. To evaluate its efficacy, researchers conducting in vitro experiment. These tests demonstrate that high concentration of NPC-31 significantly reduce the formation of abnormal protein at concentration of just 2 micromolar. It's a small molecule. Concentration. Abnormal protein will nearly eradicated in the lab setting, and PET imaging confirmed that NPC-31 did cross the blood-brain barrier, and strong evidence that NPC-31 can inhibit the disease process at a molecular level within the brain. A key aspect of support for the further development NPC-31 for the treatment of CJD emerged from studies in monkeys and mouse model of prion disease. The studies in monkeys were considered pivotal non-clinical study and demonstrate the efficacy of NPC-31 treatment before and after onset of symptoms of prion disease. Results were recapitulated in prion disease model in mice, where treatment of NPC-31 resulting in significantly longer survival than untreated mice. The overall non-clinical animal study provided important and informative data on efficacy in the form of delayed disease progression, pharmacodynamics, safety, and dosing for treatment of prion disease, which has collectively provided the foundation of advanced development of NPC-31 into clinical testing for CJD. The development of treatment for CJD align with our company's mission, contributing the society by providing necessary but neglected medicine and medical devices. In support of this goal, Nobelpharma decided to acquire the development right for this compound. We have completed several essential non-clinical studies and expected to initiate clinical studies very soon, probably next year. That's all.
You said next year.
Yeah, next year.
Yeah, as everyone introduces their potential therapeutic, if you feel comfortable, if you could also, like you just did, describe where you are in that process, preclinical versus into the clinic. So thank you very much. Appreciate it.
Thank you.
At the end, then. I'm Elizabeth McNeil. I'm with Sangamo Therapeutics. I'm a neurologist by training. I think I met some of you at the roundtables last night or yesterday afternoon. A little disoriented to time. It was yesterday. Anyway, luckily enough for me, a lot of our background work has actually been described at this conference. Earlier today, Joel, I think, talked about Sonia's grant, which was done with Sangamo. And we work with zinc finger repressors. Zinc finger repressors, if you are imagining the very nice slide that was shown where we're going to stop at the promoter for the PRNP gene in order to try to decrease substrate of prion protein. Okay, so what we do is we have animal studies in mice and non-human primates, which has shown, and actually Joel showed this data earlier today, thank you, Joel, on the survival data, which showed that we have impressive survival of the mice. I should tell you that we have studies that administered our product in those animal models at day 0, so animals that had not been inoculated with prion, as well as after 60 and 120 days, if my memory serves me properly. And so we were able to show improved survival. We also have data that shows improvement in various and sundry biomarkers. Okay, so that was really exciting. And that made us think, wow, we can go into symptomatic people. We have some data that shows that we might be able to go into asymptomatic people. FDA and most regulators say, you need to go into symptomatic people first, and then you can go into asymptomatic people because you need to know that you've got some benefit before you have risk. Okay. While that was ongoing, those animal studies, we also developed a capsid which we call STAC-BBB, which can cross the blood-brain barrier. And so that allowed us, or gives us the feeling that we're able to administer our compound intravenously as opposed to intrathecally. Why does that make a difference? Because most people find it a little bit easier to have an injection into one of their veins rather than have a lumbar puncture. As a neurologist, I don't think that's that much difference, but other people disagree with me on that. And I see that lady looking at me like, no, ma'am, that's not the same at all. All right, fine. With our STAC-BBB compound, we have looked at brain distribution. When we say that we're going to repress or suppress the amount of prion protein that's produced, it's not as though you put it in and it suppresses it at the same level everywhere. So some of you may be familiar with the dimmer switch, which is the way I think of this, is that so some parts of the brain go down a little bit, some go down a little bit more than others. We don't suppress it entirely, so therefore you still will have some prion protein lurking around, but different parts of your brain, your different neurons will suppress it to different extents, okay? So, all right. So now, that's all I'm gonna say about the preclinical and the basic science, because I'm a clinician. So for me, I wanna talk about the trial that we're gonna start trying to do in January of 2026, or sometime first quarter 2026. That's what I'm working on right now, is writing up that protocol, figuring out sites, and figuring out how we're going to do that. It will be in symptomatic people first, because as I've said, we have to do show some benefit before I can go into asymptomatic people. But the things that I'm considering, and people who came to talk with me at the roundtable know that I asked, if I can't fix everything, because obviously you want everything fixed. I can't do that. I'm going to tell you now, I will probably only be able to fix some things. So what types of things would be the things that you would want to see most urgently fixed? And I got a lot of input from the people, and I thank you all for giving me that input yesterday. You all know who you were. We also have to have things that I can quantify. So for example, people's walking ability and falling, being able to say, my loved one's having fewer falls, they aren't having as much swallowing difficulty, things that I can actually point to the regulators and say, this is a benefit because it's a neurologic disease. In neurologic diseases, stabilizing people is considered a benefit. Yes, we always would love to improve. I think of the brain as being very high maintenance and very unforgiving. You can't, if you've had an injury, it's very hard to get back to 100%. I may not be able to get you there, but we may be able to stabilize you where you come in, which makes it important for us to be able to try to treat early, which goes back to the education part. And I'm going to tell you, because Debbie was talking about the phone calls that she makes to people. As I'm doing this development, one thing I always find very important is patients and caregivers know what makes a protocol work and not make a protocol work in terms of time commitments, in terms of things such as, getting to those sites can cause a difficulty. How can you make it easier for us to participate? So we will be talking to Debbie to try to get a panel of patients/caregivers to give us input into protocols and into, I'm looking at my time and I think I've just gone over, and into what we're going to do from there. And I'm going to stop there.
Thank you very much. Nina, yeah.
Hi, everyone. My name is Nina Oberbeck. I'm the head of translational sciences at Gate Bioscience. Gate is a biotechnology company. We work to discover and develop new medicines. We are located in San Francisco in the Bay Area. And we've been operational now about 4 years. We're a very small company. We're a startup. We currently have 35 employees, very small and mighty team. And we've been working since the very early days of the company to develop a drug to treat prion disease. So at Gate, we are developing a new class of small molecule therapeutics. And these work to selectively eliminate harmful secreted proteins from the cell. And we call this new class or new modality molecular gates. And I'll explain why in a minute. And that's also why we're called a Gate Bioscience. Many people have asked that in the roundtables. So you've probably heard quite a bit over the past few days about the different therapeutic hypotheses out there for treating prion disease. And probably the most prominent one that's been discussed is the prion lowering hypothesis. This idea being that if you can lower levels of normal PRP or PRPC in the brain, then you're eliminating the substrate by which prion protein aggregates and propagates throughout the brain. And in theory, with this approach, you can kind of stop the disease in its tracks. And this is a therapeutic hypothesis that would apply both to symptomatic, sporadic CJD patients as well as genetic carriers of the disease, and can be used to treat symptomatic patients after symptom onset, but also in theory to treat pre-symptomatic carriers to prevent them from ever developing the symptoms in the first place. So a widely applicable therapeutic hypothesis. And this is also the hypothesis that Gate Bio is pursuing. So we are developing small molecule molecular gates to lower prion protein levels in the brains of people. So how do molecular gates work? Hopefully by giving an analogy, I can explain it well. So if you think of a cell is producing tens of thousands of different proteins all the time. And a subset of these proteins, around a third of these proteins, their destination is outside of the cell. They're what are known as secreted proteins. And PRP, or prion protein, is also a secreted protein. So it is made inside the cell, but it makes its way to outside of the cell. And when these secreted proteins are made, the cell knows that their destination is outside, and they get put on a special conveyor belt inside the cell. And this conveyor belt traffics them outside of the cell. And PRP is no exception. It gets put on this conveyor belt as soon as it's made. Now, molecular gates can gate the entrance to this conveyor belt. So these are essentially small molecules that allow us to selectively prevent prion protein from ever getting on the conveyor belt. And at that point, it falls out and it essentially falls off the conveyor belt and onto the floor of the factory, if you want to keep going with the analogy. And the cell says, wait a minute, this protein should be outside. It should be on this conveyor belt. It's not. It's in the wrong place. Let me try to get rid of it. And so the cell takes prion protein to the trash can of the cell and essentially chops it up and recycles it for parts. And so what you get is a functional elimination of prion protein. And that's how molecular gates work. In industry speak, or in kind of the researchers speak, we call these drugs degraders. And there have been many degraders developed for oncology indications. Usually these are aimed towards intracellular proteins, proteins that stay inside the cell. What Gate is doing that's novel is aiming degraders at secreted proteins, including prion protein. And it's worth mentioning that, I've said this already, that molecular gates are small molecule therapeutics. And small molecules tend to have quite a few advantages over other therapeutic modalities. The main one that most people think of is that they generally can be taken orally as a pill. So what we're looking to develop at Gate is a small molecule that you can take from your medicine cabinet in a pill bottle and take maybe once or twice a day from the convenience of your own home. And I think it goes without saying that that's generally considered a much better approach than having to go for repeated injections, intrathecal injections in hospital, for example. The other thing with small molecules is that they're small, and so they generally have much better brain penetrance than other larger modalities. And it's been really, really challenging in the field to eliminate proteins in the brain. This has been something that Ionis, Sangamo, and others have pioneered delivery systems to try and get drugs into the brain, but this has been very challenging, and small molecules have an easier go of it. We can optimize our molecules to make them more brain penetrant, and they also generally go evenly to all regions of the brain. And so in theory, we can get even PRP knockdown across all neurons in all regions of the brain, which would hopefully also lead to better efficacy. We've shared some preclinical data at past conferences. I spoke at the Prion conference in China. I'll be again in Brazil at the end of the year, and shared, I think, really nice data showing that our molecules can lower PRP levels in the brain by 80%. This is with once daily oral dosing for only a week. We're able to lower prion levels by 80% in the brains of mice. And we've also generated some more new data in collaboration with Eric and Sonia, and also with Joaquin Castilla, who we also heard from yesterday, showing that our small molecules significantly extend lifespan of prion-infected mice. And that's when we start administering the drug at symptom onset. So really nice survival data. So to answer Brian's question, where are we in the drug discovery process? So we are coming towards the end of our preclinical phase, and we will soon be starting toxicology studies with our molecule. This is a very important process mandated by regulators, including the FDA, that all molecules have to go through extensive tox studies in rodent and non-rodent species before being allowed to go into humans. And we hope to enter the clinic in 1 to 2 years from now. And just lastly, a comment, we wanted to mention that Gate has been a recipient this year of a CJD Foundation research grant. And we'll be using that grant money to do more efficacy studies in collaboration with Sonia and Eric's group at the Broad. I've been really overwhelmed and just incredibly honored to meet the family members and individuals who have contributed money to that grant. It's been a real highlight to meet you and talk to you all. Thank you so much from the bottom of my heart, and I'm excited to come back next year and present some progress from those studies and show data to all of you. So thank you.
Thank you very much, Nina. Eric.
I'm Eric Minikel. So I think most people know, but my wife, Sonia Valab. Sonia, would you mind standing up? So we lost Sonia's mom to genetic prion disease. She has the mutation. We changed our careers to become scientists in order to devote our lives to a cure. And we now run a lab of 14 people at the Broad Institute in Cambridge, Massachusetts. So it's an independent research institute. And we run an academic lab there dedicated to advancing prion disease therapeutics. And I'll also call out Vanessa and Leo, who are members of our lab who are attending today. Would you guys stand up? Cool. So Sonia and I have been at this 13 years now, and early on, we realized we needed to focus on PRP as the drug target, because any which way that we study prion disease, PRP turns out to be the answer, right? So we can do neuropathology and look at what is going on in the brains of people who died of prion disease, PRP, right? We see deposits of PRP. Biochemical approaches, how do we purify the so-called infectious agent that's present in animal prion diseases? It turns out to be made of PRP. We study animal models, we study human genetics, what are the risk factors for prion disease, what causes genetic prion disease? The answer is PRP. So this target, PRP, deserves most or possibly all of our attention in drug discovery. No other target that you could imagine going after in your body has even 2% of the amount of evidence that it's going to be relevant to prion disease that PRP has. You have to go after the main thing in this disease. So then within going after PRP, our strategy is to lower it. And we have really good animal models in prion disease. Other neurological diseases don't. Mice don't get Alzheimer's. They do get prion disease, right? We have really good animal models, and I believe that means we should use them. So we should invest most of our effort in therapeutic approaches where we have studied it every which way, robustly in many different experimental paradigms, in many experiments, in many different labs have replicated it to show that lowering PRP is helpful. So in our lab, we have shown this for ASOs with Ionis, for a divalent siRNA that I'll talk about in a minute. We've shown it for GATE with their small molecule. We've shown it for the zinc finger repressor with Sangamo, and we've also shown it with base editing using CRISPR. So any which way that we lower PRP, we find that that is helpful, and we know a lot about it, right? We've shown that it works across different species of animal. It works against different strains of prion. It works at different stages of disease, albeit to differing degrees. And it can either delay onset of disease if you start early or slow progression if you are not able to start treatment until later. So having all of those data from animals gives us a lot to stand on in terms of how we think about designing a program to try to do this in humans. And the other advantage of PRP lowering is that we can measure it, right? So when we dose a person with a PRP lowering agent, the hope is we can then measure in their spinal fluid to show that we lowered prion protein. And that gives us the ability to get an early signal of whether the drug is doing what we designed it to do, right? We don't have to wait years and hundreds of people to see if we modified clinical course. Even in just a few patients, we could say, did the amount of the protein go down in the spinal fluid? And that would give us a hint and help us think about, did we dose high enough? Do we need to adjust dose levels, et cetera? Being able to look at PRP lowering also gives us a way to read out that the drug is having an effect in a pre-symptomatic individual, if, as I hope, we eventually get to preventive trials. Whereas other strategies, it's harder to think about what does that do in a person who doesn't have misfolded prions in their body yet. PRP we can lower even in someone who's not sick yet. So PRP is our target. PRP lowering is what we want to do to it. And this idea is so good that it deserves many shots on goal. So unfortunately, drug development is really difficult. Plenty of things that look good in a laboratory setting will never get to a first human dose. And of things that get to a first human dose, most of them do not end up becoming an FDA-approved drug that people are taking, right? Drug development is just really difficult. It's difficult for scientific reasons. It's difficult for cost reasons. It's difficult for regulatory reasons. And a lot of things fail. And that means that we need to be taking multiple shots on goal. And in our lab, we are thrilled that we've had the chance to work with amazing people. We've worked with Ionis, we've worked with GATE, we've worked with Sangamo. These have been really successful collaborations, and I'm really excited that we have these companies on board thinking about how to develop drugs for our disease. We have also invested in trying to develop drugs ourselves out of our own lab without a company sponsoring them. And so I'll say a little bit about one of those that I hope is relatively close to clinical application. So we have worked with UMass Medical School on a divalent siRNA drug designed to lower prion protein. There's no company sponsoring this. It was an academic collaboration. And the data just looked too good to sit on and say, oh, that'll just be a mouse study. We don't want to bring it to the clinic. The data looked really good in a humanized mouse. It was potent and long-lasting. And we said, we've got to find a way to give this a chance in the clinic and see whether it works. Now, the nature of the drug, it is a chemically modified short oligonucleotide drug. So in some ways, it is similar technology to an ASO. They both lower the PRP at the RNA level so that less of the protein is produced. This divalent siRNA would also be administered by lumbar puncture into spinal fluid, just like an ASO. So they have similarities, but they're distinct drugs. And like I said, we want many different shots on goal because drugs fail for all sorts of reasons, right? They can fail on toxicity. They can fail because they were dosed too low, dosed too high. Clinical trials get designed wrong, all kinds of things. We want multiple chances here. So we were really lucky to be able to obtain NIH federal funding for developing this divalent siRNA. So in 2022, we had identified the drug candidate that we thought was promising. We got an NIH grant as well as some philanthropic support. We were able to take that through manufacturing. as well as toxicology studies in animals. The toxicology was very clean. Even at very high doses, we didn't see any adverse effects of just a single dose, which is what we've been able to do so far. We don't know anything about chronic dosing. And the status today is that as of February, we submitted an application to FDA, and we heard back in March that they have cleared us to dose symptomatic patients with a single dose. So I just want to emphasize how limited this is, right? This is a small trial. We're talking about probably 15 patients. Symptomatic only. FDA does not yet want to let us treat pre-symptomatic, even though I would like them to let us do that, and we will keep pushing on it. They have not given us permission to do that yet. Only a single dose. We don't yet have permission to administer the drug chronically. So at this point, we have clearance from FDA. The stage today is that we are waiting for NIH funding to run that clinical trial. We applied. We're currently in budget negotiations with the federal government over it. And I hope that that means that they are very likely to end up funding it at some level, right? So it may not be the full amount of funding that we applied for, but usually you don't get to this stage if there's not a pretty good chance. So responding to Brian's request that we say a little bit about the current status, again, like this is a huge knock on wood and grain of salt kind of thing, but I have been told by people who've worked in this sort of NIH network of clinical trials before that from the stage we're at today, it may be possible to dose a first patient by end of the year, but that is not at all guaranteed, and it's scary for me to say because we could not get the funding at all, or there could be various delays. Anyone who's worked with federal government knows that things are not always quick and efficient, but we are doing what we can. So I'm happy to talk about divalent siRNA as well as all of these other programs that I'm really excited about. Thank you.
Thank you, Eric.
I think it's my turn.
I think it's your turn. Yeah.
All right. Hi, everyone. I'm Ann Smith from Ionis Pharmaceuticals, and I regret I didn't have notes because as all of these lovely colleagues were speaking, I was thinking of really great things to add. It's amazing to me how collaborative this work is and this community is. Eric talked about shots on goal, and Debbie, I'm thinking about a slide you should have on your website. We have the advantage with prion disease, and I will get back to Ionis in a minute, but we have this advantage with prion disease of knowing what our target is. And we can look at that DNA to RNA to protein path and know we want to lower the protein and think about the different ways to attack that at the DNA level, at the RNA level, at the protein level. And we have just a really nice group of different approaches here, different shots on goal, which absolutely in drug development is necessary to have a success, but my gosh, imagine choices for patients too. So I love all of this that's going on, and we support each other, and it's really been lovely. Let's see. Okay, so I'm at Ionis, which when I started 10 or 11 years ago was about 350 people, so relatively small. We're a little over 1,000 now. And Ionis was formed in 1989, I think. With the sole purpose of developing ASOs. So this concept of you ought to be able to create a small little string of oligonucleotides that binds to an RNA, and by doing so, triggers a natural response in the body that's either going to upregulate how much protein is made or downregulate it. And depending on the chemistry and how you design your ASO, you can pick. So in prion disease, what we want to do is lower protein. So we've designed Ion 717, which is a little string of oligonucleotides, and it binds to the prion mRNA only, specifically, sensitively. So it's going to drive down, it's going to cause that RNA to be chopped up, which means the protein isn't formed. So we're approaching it from that really biologically simplistic approach of let's just prevent the protein from being made. Now, as you heard on the panel, it's not going to be every cell, and we're not going to catch every RNA before it becomes a protein. But we know from these really nice mouse experiments over the years that if we can just lower it somewhat, we can absolutely create a benefit in mice. And now the question is, can that benefit translate to humans? So we have had the sheer pleasure of starting a clinical trial. We announced it at the end of 2023, and over the course of 2024, and fully enrolled our first in-human clinical trial with Ion 717 in symptomatic prion disease patients. The collaboration with the community, the physician community, the patient community has been outstanding. It is amazing how you guys rose to the occasion and agreed to participate in research. So I'm at Ionis. I'm really proud to be there. I think Ionis has a long history of quality scientific work. There were probably 2 decades before we really made the core strides in antisense oligonucleotides to have them be drugs. And now there are a few dozen ASO drugs on the market, a lot of patients being treated. A number of those came through Ionis, either to a partner or wholly owned Ionis products. It's a wonderful place to be fulfilled from a scientific perspective, but I have to say it doesn't hold a candle to being here, to being in this room with the energy and the intensity and what you bring to us, the feedback directly from patients and families about what you want in a drug, what you need in a drug. We just can't replace that. So thank you all for being here and for participating in research however you do it, whether it's participation in a clinical trial or being on a panel or responding to a survey. It all matters. Truly, it matters. It helps us be on a path to bring to you an agent that might be what you want, to help you, to help your loved ones. I feel like I am rambling a little bit. What else did I need to say? Okay, so we are in clinical trials. This is clinicaltrials.gov. Is a repository you can go to look for any clinical trial that's actually open. Again, clinical trials, plural, clinicaltrials.gov. This is our first in human trial. We expect to get to a point where we will become unblinded, which means at this stage, I don't know anything about how it's going. I follow the patients and the sites, and we keep track of the execution of the trial, but we don't have data that tells us yet anything about how the drug is doing. It's a first-in-human trial. It's small. It's short. It's designed to help us understand how the drug's behaving and get a first look at safety. It's not designed to tell us whether the drug is effective. That would come later. But right now, we are just intending to learn enough about our drug to design those later phase trials. So we have our eye on the ball. We want to get to the end and answer the question, does this drug help people? And to do that, sometimes we need to do short, small studies first. So we would expect by the end of this year to have some sense of what the data are telling us, and we'll come to all of you to help you understand that as well. At that point, what happens next is going to be a function of the data. Anything's possible. We could learn that it's not safe. We could learn that the participants are getting worse on the drug, which would be devastating, but is also a data point and helps us all move forward. That's not the end. We could learn that, yes, we're seeing something interesting, but maybe based on the amount of drug we see in participants over time, we need to dose more frequently or less frequently or higher or lower. We might need to do a bit more exploration of that type. Or it could be we just really hit it well out of the park right from the start. These are the data we need to move to that next phase, to really start to test, is this a drug that benefits patients? So it's an exciting time. It is annoying to be in the dark, that I don't get to see the data as they come through, but we're coming up on it. And that's, again, thanks to the network of clinicians, some of whom are in this room, one of whom's at the table, who really partnered with us and collaborated with us and helped us design a quality trial to really do justice to the participants who are giving of themselves to join and take on this risk with us. I think that's probably it. Yeah? Okay.
Great. Thank you. And I think as a community, we should all be, of course, appreciative to everyone at the table, but especially Ionis being the #1 industry-sponsored study, first truly international prion disease therapeutic trial, as well as the first lowering of prion protein. So there's a lot of firsts. It opens the door for a lot of future things. If you remember Dr. Watt's earlier grant review talk, he talked a little bit about how science works. Science is a process, and we have to learn from failures. Most clinical trials are not successful, but you learn very valuable lessons. There have been several clinical trials in prion disease. They've been small, usually local, but we've learned a lot from them, even though they weren't successful. So again, no matter what happens with these studies, we will learn and we will get better, and that will help in the future. If you're interested in knowing about currently enrolling clinical trials, of course, clinicaltrials.gov is the place to go. If the not-so-friendly government website is not your thing, the Prion Center, as well as the CJD Foundation website, also has currently enrolling treatment trials that you can go to as well, if that's easier for you. So in summary, for today's session, we have multiple partners that are willing to work with us, and many of them are very close or actually in clinic. 4 of them are actually lowering the prion protein itself, not 100%, but lowering it significantly in most cases. And then we have another approach that makes the prion protein stable and a little bit more unable or less likely to misfold and cause problems. So if you look at most diseases are not treated with just one treatment. Take high blood pressure, for example. A lot of people are on more than one anti-blood pressure medication. Same thing for cholesterol. Alzheimer's disease now has 3 different therapeutics that you can take concomitantly. So again, we hope for success in all of these studies, and it may be that we use multiple drugs at one point.